and I'm happy to introduce him to you today. Art graduated from the University of Illinois with his PhD in 1974, and he's been here at Emory for over 30 years. He's a fantastic mentor, and he recently joined the ranks of Emory's 1% after getting a perfect score on one of his grants. So he'll be talking to you today about performance.
Thanks everybody for coming on a really nice day and sitting in here rather than outside. As Laura indicated, my lab studies ways to enhance axon regeneration in peripheral nerves. And the reason we do that is on this slide. This is our epidemiology slide. Some of you have seen different versions of this, and you can take a nap now if you want. We study this because peripheral nerve injuries are common. There are literally hundreds of thousands of new traumatic peripheral nerve injuries in the U.S. every year. And if you believe the patient advocacy websites, there are at least that many that are not reported. The common perception out there among clinicians is that peripheral nerve injuries heal themselves. And this is unfortunately not the case because fewer than 10% of patients with traumatic peripheral nerve injuries ever recover full function. And the thing that's usually blamed for this is that axon regeneration is slow and inefficient. Not all the axons get started at the same time. And in the process of regeneration, some of them regenerate more slowly than others. And part of the problem is that the pathway through which the regenerating axons have to grow to reestablish connections with their targets in the periphery is replete with both growth promoting and growth inhibitory molecules. And over time, the growth inhibitory molecules win the battle so that if regenerating axons have to grow a long distance, they grow through an increasingly hostile environment. And so the premise of our lab is that if we enhance axon regeneration, we might be able to improve functional recovery. What I want to do today is introduce one way that we've studied for enhancing axon regeneration, and that's with exercise. And tell you a little bit about a story that's relatively complete, and then tell you 3 stories that are at different stages of completion that are ongoing in the lab. So the first thing I want to do is show you the model system that we use. People that hang out at the Neuroscience program website have probably seen this pop up because it pops up an embarrassingly large number of times when I'm on that site. This is the L4 dorsal root ganglion from a 5-1-YFP strain H mouse. And in this mouse, yellow fluorescent protein, a spectral variant of GFP, is expressed under the control of a promoter for Thy1. That targets it to neurons, and it's found both in these beautiful dorsal root ganglion cells and in the axons in the dorsal roots, but it's also found in motor axons in the ventral root. This is the periphery out in this direction. And you can see that it's not in all of them, it's only in a subset of them. And that's actually an advantage to the way that we do the experiment because we think that the marking of axons in this mouse provides a sample of all the axons in the peripheral nerves. The general experiment that we do, I'm going to show you a number of variations on the theme of this experiment, is this one. We start with this mouse as a host, and we cut one of the branches of the sciatic nerve. In this case, it's the common fibular or common perineal nerve. Sometimes we've done this also with the tibial branch here. Then we repair it, not necessarily by an end-to-end anastomosis the way that you would do clinically, but we use a graft, a piece of the same nerve, from another mouse. Almost always this is a littermate of the mouse that we use. It's non-fluorescent. Our original intent in doing this was to provide a nice dark background against which we could see these beautiful yellow axons regenerating. But over the course of doing these experiments, we realized that this could actually be a very cool experimental tool because we could now use mouse genetics to manipulate both the environment surrounding the regenerating axon by manipulating the donor versus manipulating the host. And I'm going to show you some experiments in which we've done this. So we cut this nerve, we graft on a hunk of another nerve, and then we secure it in place with fibrin glue. The fibrin glue also gives us an opportunity to experiment with this model because we can put various growth promoting or growth inhibiting molecules into the fibrin glue. We then let the animal survive 2 weeks, and we harvest the nerve, and we can, these axons are so bright we can use a whole mount and make confocal optical sections through it, and then simply measure the length of the regenerating axons in reconstructions of those sections. And this is what you see here. This is a cumulative histogram with cumulative percentages along this axis and axon profile length along this axis. And I'm going to show a bunch of these today. These are the data from a control animal. The size of the symbols here, or the bubbles, is proportional to the standard error of the mean. In all cases, I'm going to show you an N of 4. And so bigger bubbles means more variability. What we're going to derive from these very often is what is the axon profile length here at the 50th percentile. Maybe it's crapping out. And you can see that in untreated animals, it's about 1 millimeter, 1,000 micrometers in after a 2-week survival period. So one of the first things that we experimented with was trying to look at treadmill training. And the reason we got started with this, at least is how I remember it now, at the point we started this, there was a lot of buzz out of Reggie Edgerton's lab, out of treadmill training as a way of improving functional recovery after spinal cord injury. And so we put animals on a treadmill and we asked them to walk, and then we repeated the experiments, and we found this enormous increase in axon profile length here. You can see this is the 1 millimeter of growth in 2 weeks, and this is what you get with 2 different exercise paradigms. You get more than doubling of the axon profile length. And so the axons, the regenerating axons, have elongated more than twice as far during the first 2 weeks. We used 2 different paradigms here. We started with this paradigm, which we call continuous, because that's what Reggie's group had done, 60 minutes at a really, really slow speed for a mouse on the treadmill. And they walked continuously. And then Dario Carrasco, a former postdoc in my lab who now works with Marty Pinter, raised the issue of, is that the way that mice would run if you put an exercise wheel in their cage? And when we looked into that, we found that they don't run that way at all. But in fact, what they do is run really, really fast for 2 minutes, then they rest for 5 minutes, and they just keep doing that over and over and over again. Some mice running as much as 10,000 meters a night. At the time, Manny Savache, this person here, was doing the work in the lab. He has a background in exercise science. He looked at this and he said, oh, that's interval training. And so we tried interval training, and it worked very nicely when they ran the same distance as they did, the 600 meters, with the continuous training. And then we, because of my perspective, I said, ooh, let's see if we can shorten this and reduce the number of repetitions. And we found that 4 repetitions, 8 minutes of exercise in this interval training paradigm worked as well, as you can see here, as the 60 minutes of continuous walking. So we had an effect, and at this point we wanted to try to figure out what was the basis for this effect of the exercise. And the stuff that was coming out of Edgerton's lab at the time suggested that it was brain-derived neurotrophic factor, and they'd shown that exercise increased brain-derived neurotrophic factor expression in the rat spinal cord. And so we looked in the mouse spinal cord, and with the help of Gary Bissell and Mei Zhu, who was a postdoc in his lab, and Tiffany Zhang, who was like, oh, I love this picture, she's looking so thoughtful. We did some in situ hybridization on spinal cords from animals that were unexercised here or exercised here. These were literally run on 2 slides in the same run. And what you can see is that the exercise does produce an enormous effect on BDNF expression in the spinal cord. It's in big neurons like these that we think are alpha motor neurons, but also a whole bunch of other neurons as well. And this was all really interesting, but it became a little more complicated because it had been known for some time that both regenerating axons in the proximal stump and the Schwann cells in the distal stump, the Schwann cells that are surrounding the axons while they're regenerating through the distal stump, contain neurotrophins. And the conventional wisdom in the peripheral nervous system at the time was that BDNF in the distal stump was providing a retrograde signal to the regenerating axons and causing them to elongate. And this is because the regenerating axons, but not the Schwann cells, contain the full-length TrkB receptor. So we had to figure out a way to try to sort out where the BDNF might be coming from if it was involved in the process. And this was work that Jen Wilhelm did in the lab when she was a postdoctoral fellow. And what we're showing here is if you knock out BDNF expression in the graft, in this case, in what we think of as the distal stump, by using a mouse that expresses pre under the control of the promoter for ciliary neurotrophic factor, it knocks out BDNF selectively in Schwann cells. So here is a picture from a control animal. Here's a picture from a control animal here. You can see the axons grow about that far in 2 weeks. If you, instead of using a graft from a wild-type mouse, you use a graft from one of these Schwann cell-specific BDNF knockout mice, the axons don't grow through there at all. And this would really support the sort of conventional wisdom that there was a Schwann cell producing a retrograde signal that was driving the regeneration. If you apply BDNF, recombinant human BDNF in the glue that we use to secure this graft in place, you could not only reverse this, but you got this enormous stimulation of regeneration. But if we could also get the same thing if we exercise the animals. So all of these, except this one, the graft has no BDNF. The only source of BDNF is coming from the organ. This is what you see when you measure the axon profiles. You see that there's this big shift to the left in the red dots here. The gray dots here are the controls, the untreated animals. And you get a big shift to the left when you knock out BDNF in the environment surrounding the axons. And then this is the effect of the exercise, and the green is the effect of adding recombinant human BDNF. Sorry, I said that wrong. These are both effects of exercise. One is with CNTS3 knockout graft, and the other one is with a wild-type graft. So the point of this is that the effect of the exercise seems to be independent of the BDNF content of the environment surrounding the axons, in this case, the distal nerve segment. So from this, we proposed that it must be the neurons, right? And so Jen did experiments where we made conditional BDNF knockout mice. These are mice that we started with a mouse called SLIC, which stands for single neuron labeling with inducible pre-mediated knockout. And I wonder how long Josh Sains' lab spent trying to figure out how they could use SLIC in this game. It was at least a whole afternoon, I'm sure. These animals express tamoxifen-inducible Cre and YFP, just like the animals that we've had before under the control of the Thy1 promoter. And then when we bred them with floxed BDNF mice, we could generate a knockout that was neuron-specific. And when you do this neuron-specific knockout, what you find is this big shift to the left again. So this is a knockout host with a knockout graft. All of the data in this graph are from exercised animals. So in exercised animals, if neither the host nor the graft has BDNF, then there was no effect. If the host was knocked out, but there was BDNF in the graft, then regeneration was relatively normal. And if you use heterozygous animals, which is what these other two are, you can see that there is also only a little bit of BDNF is required. And you can see here, these are the medians now. This is what you get if you knock out BDNF in the neurons. With exercise, there's no effect. And if you have it in the distal stump, you do have an effect. And so from this, we concluded that there are two neurotrophic mechanisms here that are regenerating axon growth in peripheral nerves. One is the traditional retrograde signal from neurotrophins, principally BDNF, but we think maybe NT4/5 also. This is produced by Schwann cells in the pathway used by the regenerating axons. It stimulates neurite outgrowth by signaling through the TrkB receptors on the growth cones of the regenerating axon. We think that normally this is the dominant mechanism because if we block it in the knockout experiments, the regeneration is blocked. But we think there's also an autocrine signaling mechanism where regenerating axons secrete BDNF, and this stimulates the elongation by binding to TrkB receptors on the same neurons. Normally, this doesn't compensate, as we've seen, but it can be brought into play by exercise. So we think that the effect of the exercise is to induce this autocrine, this second BDNF signaling pathway. Okay, I should ask if there are questions so far. I feel like I'm on an express.
Can you repeat some of that?
Which part? So I want to go over the first story, and that is that exercise makes a difference in males and females. This is Kylene Wood. Kylene's now finishing her second year of medical school at the Baylor College of Medicine. And when she came in the lab, we gave her the paper that Dario gave us. And she pointed out that when animals have a wheel in their cage, the females run nearly twice as far as the males every night. And she asked me, have you ever considered that there might be a sex difference? And my response was, well, no. But if you go and look in the book, in a lab notebook, you can tell. And so I'm embarrassed, but not so embarrassed that I won't say in public, that when she looked at this, all of the animals that we had done with the slow, continuous walking were males. And when Kylene did these in females, she found that it doesn't work in females. So this is median axon profile length. These are untrained animals showing that there's no sex difference if you don't do anything. But if you exercise them by having them walk slowly on a treadmill for an hour a day, 5 days a week, you get this enormous increase, 2.5-fold increase in axon profile length, and absolutely nothing in the females. She did the obvious, simple experiment by castrating the animals, and castrating the animals makes the effect go away also. So in castrated males, you don't see any effect of this slow, continuous training. And this suggested that on top of all the stuff I've just told you about BDNF, there was also some sort of androgenic component to it. She then went on and looked at the interval-trained females and found even more interestingly that in the interval-trained females there was an effect, but in our data set we had only one male and all the rest of them happened to be females. When she did more males, she found there was no effect of interval training in the males. So males respond well to slow continuous walking, females respond well to the interval training, but not vice versa. And so this considerably muddied the waters for me at least. And then a second student in the lab, Kevin Liu, asked, well, what happens with exercise might be that you're somehow manipulating the metabolism of the androgens in the females with the interval training. And so he treated a bunch of unexercised animals with anastrozole. Anastrozole is an aromatase inhibitor. P450 aromatase is an enzyme that normally converts androgen or androgen precursors into estradiol. By blocking it, we reasoned that you might have more testosterone available in the females, and when he did these experiments in the females, he found a really large increase, almost 3-fold increase in the length of the regenerating axons 2 weeks later. So there's a sex difference, and we think that it's somehow tied to androgens.
I'm sorry.
Yeah, go ahead.
Previous slide, is that continuous training in females? Is that
There were these were untrained,
Okay.
completely untrained. All we did was block aromatase. So more recently, Nick Thompson in the lab, along with our collaborator Dale Senglob at the University of Indiana, I guess it's Indiana University, have investigated this further. What they've done is developed a sustained release device. It's a silastic capsule filled with flutamide. Flutamide is an androgen receptor blocker. It binds to the androgen receptor and prevents native androgens from binding to it. Depending on who you read, it's alleged to not have any endogenous activity from binding to the androgen receptor. when Nick did this, he found that the weight of the seminal vesicles in the males was dramatically decreased, as was the weight of the antigen-specific bulbospongiosis muscle. But the more remarkable thing that he found was that treating these animals that were appropriately trained, either continuously for males or interval training for females, blocked the effect of the exercise. So the black lines here represent the untreated controls. I didn't put bubbles because it got too busy. The gray bubbles here are from castrated males. The pink is the flutamide-treated animals. Notice that they're not really significantly different at all from the intact. And the purple is what you've seen before. These are the effects of the training in untreated animals. And you can see from these bar graphs of median length that the only two that are elevated are the males that were not flutamide treated and the females that were not flutamide treated but were exercised. There's no effect of flutamide by itself. This thing's driving me crazy. Here and here. So we think that the effect of exercise has to then rely on androgen receptor signaling in both the males and the females. Yes, Don?
Just a general question, Art. In treadmill training, you have a general metabolic effect on lengthening muscles. If you did an experiment where you stimulated the proximal stump of that nerve, would you find the same thing? Other than exercise?
Yeah, but I have two responses to it. The first one is a short response, yes. The second one is, wait a couple slides and we'll show you. Yeah, I want to point out that at least for the continuous training, it's very slow. It's a couple of recent papers have shown that the speed at which we walk these animals is well below something called a lactate threshold, which is a threshold for the induction of all sorts of things like cortisol. I don't think it's true here. I think these animals are probably stressed. But notice that this doesn't work in the males, so it's still a bit of a mystery. So what we want to try to do is figure out where this is all happening. And so I've begun a collaboration with this guy, Jeff Zajac. He's at the University of Melbourne in Australia. And Jeff is one of only 2 people in the world, but the only one who's not retired, who have made a flocked androgen receptor mouse, a mouse that we can use to make conditional knockout mice. And in Jeff's mouse, exon 3, which is a critical DNA binding domain of the receptor, is flocked, and we can cut it out using the Cre-Lock system. The androgen receptor is a ligand-gated, acts like a ligand-gated transcription factor. Exon 1 binds the androgen, and exons 2 and 3 bind DNA after the receptor ligand complex is translocated to the nucleus. And so we're hoping that we can knock this out. And I could talk for a very long time about the fun of having to try to import mice from Australia. It's taking a really long time. What we want to do with this is use 4 different Cre mice, a tamoxifen-inducible Cre that will knock out the androgen receptor in all cells. And what we should do is mimic the tamoxifen experiments. We can use the slick mice that we've talked about before to do a neuron-specific, but we can also use 2 different mice, either a chat Cre mouse or an advilin Cre mouse, to knock the androgen receptor out, predominantly in the motor neurons. Until yesterday, I could say exclusively in the motor neurons, but I was educated yesterday. And advilin Cre is found only in sensory neurons, in dorsal root ganglion cells. And this is work that Joe Ward, a new postdoc in the lab, is going to be working. Okay, so that's my first short story. The second short story is, what is it about exercise anyway? And we know that BDNF production by the neurons is required. That's what Jen Wilhelm's experiments showed. And early observations, and I'm being sarcastic here when I say all the way back to 2000, showed that increased neural activity is associated with walking. That is associated with walking is what drives the BDNF expression in the neurons. And in vitro, neuronal BDNF expression has been shown by several groups to be activity dependent. Our contention was, well, maybe it's the activity. And what caused me to think this was a series of experiments that Manning Sabatier did while he was in my lab that we still haven't finished. And he actually had animals walk on a treadmill that was inclined up at 20 degrees. When you do this, it obviously means much more work for the animals. And there's a recruitment of motor neurons that probably weren't recruited even during the fast level walking. And we assayed this by retrograde labeling from both the common tubular and the tibial nerve. And you can see that with level training, we get a modest increase in the number of retrogradely labeled cells 2 weeks later, but we get a much larger increase when we're walking upslope. And so these were all consistent with this original idea going all the way back to 2000 that it might be the activity. However, more recently, some of the very same people who were saying that it was the activity stupid, well, they didn't think stupid, but it was the activity, are now saying that it might be other things like environmental enrichment. You know, you just feel better when you've been exercising. Oxygenation state, one of Sean's favorites. Not really, right? The neurons might become slightly hypoxic while they're walking, and that might have an effect. Caloric utilization has been invoked, even diet, and oh yes, testosterone. So we wanted to test the idea, or at least begin to test the idea, that it was the activity that was responsible for the enhancement of the increased activity in the neurons was responsible for the enhancement. It was also fueled by a study in around 2000 by Tessa Gordon's lab showing that as little as 1 hour of continuous electrical stimulation at the time that you repaired a cut nerve would enhance the regeneration of the axon. And so we've turned to doing this without the genetics. Now, the genetics, I really wanted to show this slide simply to show this rat because it's such a cool picture. This is from Carl Diceroth's website, which has become sort of the user's manual for people doing optogenetics. But optogenetics is a way to integrate optics and genetics to in this case, achieve gain or loss of function. This is a quote from Yizar et al. And the idea here is microbial opsin genes can be introduced into cells, and then you can control the activity of those cells by shining light on them. And you can use the genetics to target the light-sensitive probes, light-sensitive ion channels, essentially, to particular neuron types. The most common one is the one that we've used is channel rhodopsin. It's derived from Chlamydomonas. It's a light-activated cation channel. It's not particularly specific. It opens under blue light, and it's in the cartoon here, and it allows cations to go inside the cell from cellular space. There are two inhibitory opsins. These are known as halorhodopsin, which is a chloride pump, or archirhodopsin, which is a proton pump, and these are activated by yellow light. That's what is shown in these two cartoons. And they produce a hyperpolarization of cells, and they've been used to silence. This field is moving really fast, and there are now all sorts of genetic variants of these opsins that can achieve different sorts of effects. For example, there's an opsin known as cheetah, which has a very rapid onset. There are step opsins, which are known to depolarize neurons and keep them depolarized until you turn them off. There are new ones coming out all the time. So how have we used them? Well, this is work that Laura is doing in the lab. And this is a picture of Laura holding a fourth-letter poster at the Society for Neuroscience. And I want to point out that you can see Laura, except it's a little blurry here around her hand, because she's doing so much of this. We've used a mouse that just kind of fits right into our general model thing. This is a thigh 1 channel rhodopsin YFP mouse. It has the same sort of construct as the one we've used before. This is a dorsal root ganglion from one of those animals. And if you remember the dorsal root ganglion I showed you on the first slide, there are a lot more cells that are yellow on this one. But this is just to show that we have both sensory neurons and motor axons that have the channel rhodopsin and YFP. If we look out at the neuromuscular junction, we see that there are some of these that are So we have alpha-bungar toxin binding sites marking the postsynaptic receptors on muscle fibers, and only some of these have the YFP in them also. We've estimated actually that it's about 40%. If we shine blue light on these with really small LEDs, then we can cause these muscle fibers to become, or these neurons to become active, and they'll drive muscle fibers. These are EMG recordings made from an animal where we stimulated with trains of stimuli at 20 hertz, and you can see that they follow the train pretty well. And Laura liked to point out that there's also no stimulus artifact here. When we've done this, when we've compared electrical activation of these of the same muscle to optical activation of the muscle, we get these curves. These are adjusted so that the EMG response is plotted along this axis and the stimulus intensity in multiples of threshold along this axis. And you can see that these curves are really, really quite different. Based on our evidence and evidence from Scott Delp's lab at Stanford, it's now pretty clear that you activate the small motor neurons first when you stimulate with light rather than the opposite direction when you stimulate them electrically. This is probably because the density of the channel rhodopsin of the transgene in the smaller cells is higher than the density higher in the larger cells, and therefore you shine light on them, the first ones to be activated are the small ones. The other thing that's useful, that will be useful to us, is that there's a much larger range of activation intensities. We can grade this much more gradually, which we think is a way of trying to mimic the effects of different intensities of exercise. So that's okay. I just said that. Okay, so this is what we've done. So what Laura's done so far. She started with this mouse. She put a cuff up here on the sciatic nerve, and she activated the axons with blue light at 20 hertz for an hour. And then when she was finished doing that, she cut the common fibular nerve and repaired it with a graft, just like we described before. And I've got to keep up with my mouth. After 2 weeks, she harvested the nerve, optically sectioned it, and measured the lengths of the YFP profiles. And this is what she found. These are the black dots here, are what you've seen ad nauseum before.